Bonjour! Let's continue our tour through Notre Dame. We'll travel back to the year 1829 when Victor Hugo began writing The Hunchback of Notre Dame to explain how purine nucleotides are degraded and salvaged. You're probably thinking, Ricky, what does Victor Hugo and Notre Dame have to do with salvaging purines? Well, you see, the Cathedral of Notre Dame, once viewed as the pinnacle of Gothic architecture, began losing its charm by the 17th century. King Louis XIV replaced the stained glass windows with plain glass, and the subsequent French Revolution caused massive damage to the cathedral. Victor Hugo wrote The Hunchback of Notre Dame to raise awareness of the importance of preserving Gothic architecture. Like Victor Hugo, we take a stand here at Sketchy Medical, but to raise awareness on the importance of recycling purine nucleotides, at least so you can ace your exams. We spend all this effort making these nucleotides, so it's probably worthwhile to recycle them. We shall not let no purines go to waste. As we go along, we'll draw arrows on the wall of the Notre Dame Cathedral to match this diagram, showing the actual purine degradation and salvage pathways. Before we talk about purine salvage, let's discuss how purines are degraded. First, DNA and RNA are metabolized to AMP and GMP, as well as the pyrimidine nucleotides. We've drawn falling pieces of debris that look like a double helix to help you remember that AMP and GMP are made from the breakdown of nucleic acids. Remember these lamp and grump gold statues? They represent AMP and GMP. AMP and GMP are converted to IMP, shown by the IMP gargoyle. AMP deaminase turns AMP to IMP, while GMP reductase turns GMP to IMP. Note the double-sided arrows because IMP may be converted back to AMP or GMP. This happens during de novo purine synthesis, as we previously discussed, or purine salvage, which we'll talk about shortly. In the next step, IMP loses a phosphate group to make inosine, a nucleoside. We showed another similar gargoyle right below the IMP gargoyle, but this one doesn't have a P staff because it's a nucleoside. Recall that nucleosides only contain a sugar and base, whereas nucleotides like AMP, GMP, and IMP contain a sugar, base, and a phosphate group. We'll dub her I gargoyle for inosine. AMP doesn't necessarily have to become IMP in order to make inosine. AMP may instead lose the phosphate group to make adenosine before turning into inosine. You'll see a diagonal arrow connecting the first lamp statue with another lamp statue that doesn't have a P staff. The lamp statue with no P staff is adenosine. The enzyme adenosine deaminase catalyzes the next step of converting adenosine into inosine. Illustrated here is a D animator drawing a caricature of the lamp gold statue. He's adenosine deaminase. Adenosine deaminase deficiency causes severe combined immunodeficiency, or SCID. We'll talk about SCID in the next sketch. We've encountered the I gargoyle once again, I for inosine. In the next step, inosine loses the sugar group to make hypoxanthine, a free base. The free-falling hippo gargoyle is hypoxanthine. Then, xanthine oxidase turns hypoxanthine to xanthine. Xanthine oxidase is represented by the tic-tac-toe XO graffiti between the falling hippo and the man playing a xylophone. Poor guy doesn't realize a hippo's about to fall on his head. By the way, the xylophone will help you remember xanthine. Let's have another look at that original grump statue. Earlier, we said GMP turns into IMP in order to be degraded. Alternatively, GMP can lose a phosphate group to form guanosine, who will be represented by another grump statue, but this one doesn't have a P staff. In the next step, guanosine loses a sugar group to make guanine. The free-falling grump statue is guanine. Afterward, guanine is converted to xanthine. To help you remember, you'll see a rope wrapped around the falling grump statue and facing the guy playing the xylophone. Xanthine oxidase not only turns hypoxanthine to xanthine, it also turns xanthine to uric acid. We drew some more tic-tac-toe XO graffiti between the xylophone and a lady knitting a yellow ball of uric acid yarn. Gross, lady. Excess uric acid crystallizes and deposits in joints, tendons, and connective tissues, resulting in gout. Allopurinol and febuxostat treat gout by inhibiting xanthine oxidase and uric acid production. The pure nun erasing the XO graffiti will help you remember allopurinol. The XO note tucked in her pocket is febuxostat. After making uric acid, urate oxidase turns it into allantoin, which gets excreted in the urine. The rusty fountain containing yellow water is urate oxidase. We initially wanted to name the fountain Allantoin, but we decided on Atlantis, but only because it honestly just made way more sense. Rasburicase is another gout drug. It's the recombinant form of urate oxidase. It also tastes delicious, just like this raspberry. Nah, just kidding, it doesn't. Rasburicase increases the conversion of uric acid to allantoin. Okay, now that you know how the purine nucleotides are degraded and excreted, we can finally talk about purine salvage. The purine salvage pathway recycles purine bases from the degradation of DNA and RNA to make purine nucleotides. This pathway turns the free bases adenine, guanine, and hypoxanthine into their respective nucleotides AMP, GMP, and IMP. Purine salvage requires the enzymes HGPRT and APRT. Hypoxanthine guanine phosphoribosyltransferase, transferase, or HGPRT, transfers a phosphate group and ribose sugar from PRPP to hypoxanthine, making IMP. The high priest saving the falling hippo is HGPRT. Note the arrow of the rope pointing to the imp gargoyle because this enzyme turns hypoxanthine to IMP. And just beneath the high priest is a pentagonal pedestal because HGPRT requires PRPP. HGPRT deficiency causes Leishman syndrome, but we'll save that for the next sketch. Anyways, IMP eventually becomes AMP or GMP. If you don't remember the steps, please watch our purine synthesis sketch. Looks like we aren't done just yet with the high priest. He's also trying to save the falling grump statue because HGPRT can also make GMP directly from guanine. Okay, we've talked about guanine and hypoxanthine, but what about adenine? Adenine phosphoribosyltransferase, transferase, or APRT, transfers a phosphate group and a ribose sugar from PRPP to adenine, making AMP. There's a priest saving the falling lamp statue. That's APRT. Note the arrow on the rope pointing to the highest lamp statue because APRT turns adenine to AMP. And once again, there's a pentagonal pedestal hanging beneath the priest to remind you that APRT needs PRPP. Okay, it's about time to wrap up the sketch. Thanks for everything, Victor Hugo. To recap, purine degradation occurs as follows. DNA and RNA are metabolized to the purine nucleotides AMP and GMP. AMP and GMP can be directly converted to IMP and then inosine. Alternatively, AMP may be converted to adenosine before turning into inosine. Adenosine deaminase catalyzes this conversion. Inosine is subsequently converted to hypoxanthine. Xanthine oxidase catalyzes two steps, hypoxanthine to xanthine and xanthine to uric acid. We should also mention that GMP can be alternatively metabolized to guanosine, followed by guanine, before becoming xanthine. The gout drugs allopurinol and febuxostat inhibit xanthine oxidase. After making uric acid, urate oxidase turns it into allantoin, which gets excreted in the urine. Rasburicase is another gout drug that works by increasing the conversion of uric acid to allantoin. Salvaging purines requires HGPRT and APRT. HGPRT uses PRPP to turn hypoxanthine and guanine to IMP before making either AMP or GMP. APRT uses PRPP to turn adenine to AMP.